got some disclosures and then on to the talk. So I'm going to talk to you today, switching gears a little bit about the different variants and vaccines, et cetera. Talk about the condition of long COVID and what we've been learning about it. So I think many people are familiar with the fact that when you recover from COVID-19, you can transition into a state of chronic disease. And this happens for a pretty large fraction of people, probably maybe 15, 20%, 6 months out. It's not really 100% clear yet. It's not clear how it depends upon what the SARS-CoV-2 type was. But what I'll talk about today is what we found for really post-Omicron type of COVID variants. And the long COVID symptoms can be neurologic, it can be pulmonary, cardiovascular, just general fatigue and myalgia, et cetera. And they bear a lot of similarities to other conditions such as post-acute Lyme or chemo brain, post-sepsis. And so because we've had the patients that have volunteered their time and their blood to allow us to look at patients over time, we're learning about long, but I think maybe some of these lessons apply to some of these other long diseases as well. So the goal that we set out to achieve was to do really a very, very comprehensive set of measurements on the order of 300 patients over time from 2 independent cohorts, one collected at UW through Helen Chu's program and one through our own through a program that I ran through Jason Goldman at Swedish in Seattle. And to identify what we think are risk factors, which I think is a relatively small number of risk factors, some of which may be causal for long COVID. And then to associate this matrix with the symptoms that people are reporting when they've recovered. And so in our study, we're only looking 2 to 3 months out. And so I will put that as a caveat to begin with. We have longer term studies going on now, and I'm not going to comment on those aren't complete. But first we want to know, what are these risk factors? At what point in the disease course can we identify them? And are they independent or are they interrelated? So when we studied these patients, this is the time course of blood draw, one at diagnosis, one at about a week later. Typically that's when acute disease is at its peak, at least in those more severely infected patients. And then 2 to 3 months later, and we're looking at almost everything one can imagine looking at through the blood, plus electronic health records and questionnaires and what have you. And I think when you think about these types of multi-omic analysis, it's not always clear what assays are giving what information. And so I've got these various symbols, and I'll refer to these through my talk so that you understand what data is used for what. Okay, so if we look at our patients, and here I'm comparing what we found plus something that the Providence healthcare system established, what's called My COVID Diary. That's something like 20,000 patients signed up for, to basically give a diary and answer questions, et cetera, on their disease journey from acute disease all the way to recovery. We find that we're pretty much in line in terms of the most common types of symptoms that are reported. Fatigue being most common, gastrointestinal things being a little less, and somewhere in between are neurological issues such as brain fog, et cetera. If you look at our study, those that had the milder disease relative to severe infections, you find that infection severity does play a role, but it's not a huge role. It does increase your risk for long COVID, but not dramatically. Most of the patients, in fact, virtually all, but say 1 or 2 in our study, by the time that they have at this recovery stage, 2 to 3 months out, they have neutralizing antibodies. The level of those neutralizing antibodies more or less associates with the severity of the infection they had, which has been shown before. So our patient population looks fairly representative of patients across the country. If we look at pre-existing conditions in our patients by just mining through the electronic health records and patients, what we find is there's a number of things that pop up, but really type 2 diabetes is the major factor. And the reason why we resolve this effect is that with a cohort of 200 to 300 patients, you just have a lot of type 2 diabetes patients. And so it's pretty easy to resolve that as a factor. You do see issues such as coronary artery disease and other cardiac conditions being important, but those are relatively few patients. I'm pretty sure that will pan out over a larger studies, but nevertheless, diabetes is the one that really jumps out. We mined, and one of the first things we did was to mine the plasma proteome and metabolome. And the goal here was, when we had heard this from many patients, and I know this is not news to most people, is that when patients were going back to the physician and complaining of inability to exercise or chronic fatigue or what have you, not all the patients were being taken seriously. And so we wanted to maybe establish some quantitative biomarkers that could be associated with those more, more general and oftentimes more vague complaints. And so when we looked at patients that were complaining of neurological issues such as brain fog or memory issues, what have you, in the plasma proteome, and we were looking at about 500 proteins, the only thing that really came out, and it came out with high statistical signal, was a disrupted circadian sleep cycle. So there does seem to be at least some biomarkers that associate with neurological PASC. When we looked at the set of symptoms that we tended to call respiratory viral PASC, which would be chills, aches, fatigue, we found that in the metabolites, the one thing that popped out at the same time that the patients were reporting these symptoms was repressed cortisol and cortisone levels. And this is reminiscent of a condition called adrenal insufficiency or Addisonian crisis, depends on the extent of it, with very similar set of conditions to what are being reported by these patients with PASC. And so one question that is oftentimes asked when you see repressed cortisol and cortisone levels is, are these patients on steroids? And the reason why is that many steroids that patients are given are sort of molecular mimics of cortisol and cortisone and can therefore impact the body's production of those steroids. And if you look at this initial diagnosis or at acute disease, in fact, low cortisol and cortisone do in fact associate with these steroid treatments, but not at recovery. And so this seems to be something that is independent of steroid administration. And for Addisonian crisis, this is oftentimes just treated with cortisol replacement therapy. And so this does suggest, one avenue for treatment that is worth exploring, and I think is being explored. At around the time that we were beginning to work up this data, there were reports based on looking at antibody measurements in the plasma that latent viruses such as EBV or CMV might be important in patients reporting long COVID. And so one can assess these reactivation of latent viruses maybe indirectly through antibodies, but actual direct detection of the virus is much more powerful and can associate, because it's very, very transient in the blood, can associate with the event that reactivated those viruses. And so we analyzed all of our patient bloods at the various time points for both EBV viremia, CMV viremia, and for SARS-CoV-2 RNAemia. And what we found is that, in fact, it is very transient, and you can find it in patients predominantly at diagnosis. At acute disease, it's dropped by quite a bit, and it's barely detectable out 2 to 3 months later. We don't find it for CMV. There's no latent activation evidence for CMV that we find, but we do find it for Epstein-Barr virus. And when we did document it for SARS-CoV-2 RNA-emia in the blood, which had been independently shown in a paper published last year to associate with patient mortality. And so I think the level of SARS-CoV-2 RNA-emia that one measures in the blood is, in some ways a surrogate for severity of disease, but EBV reactivation is not. It doesn't really associate with severe disease at all. And when we look at those patients that were reporting EBV reactivation, what we found is that actually for both EBV and SARS-CoV-2 RNA-emia is that there were significant relationships, even when you correct for disease severity, for neurological PASC. And so the bottom line of this measurement is that these reactivation of latent EBV seems to associate with neurological PASC. Autoantibodies, I think, anybody who's been following the scientific literature at all knows the importance of autoantibodies and their impact on acute disease. And so we looked at those, but, and of course, interferon alpha type autoantibodies are amongst the most important, have been pointed out by many workers. I'll show this one paper here out of Bastard and Casanova's group, showing the importance of autoantibodies against interferon alpha for COVID mortality in older patients. But we had, about a year and a half ago, we published a Cell paper on acute disease where we had identified this subset of atypical memory B cells. And this is, in the literature, there's, I guess you would call it an ongoing debate. So atypical memory B cells are B cells that mature by an extrafollicular pathway. So it's an unusual B cell maturation route. And they are associated in the literature initially with autoimmune diseases such as lupus, but there's also been reports that maybe they're just a natural consequence of an infection. And I actually am going to point out that both are true, but they actually do associate more strongly with patients that have at least autoantibodies that are oftentimes measured in lupus patients. And so as a result of this earlier work from our Cell paper, we actually looked at a panel of autoantibodies associated with systemic lupus erythematosus as well. And what we found was pretty striking. So here's the various isotypes of SARS-CoV-2 antibodies that had developed in these patients out at convalescence. Here's the levels of these autoantibodies. And you find that if you have one of these isotypes against SARS-CoV-2, you have others. There's high correlations here. The same is true to a lesser extent, but it seems to hold true that if you have one autoantibody, you're likely to have others. But there were anti-correlations, especially with the mature isotypes of the SARS-CoV-2 antibodies with the autoantibodies. And the suggestion here is that the more autoantibodies you have, the less protective antibodies you develop. And it may just have to do with some sort of a capacity of the immune system, or it may have to do with the fact that the autoantibodies are functional and that they are actually impacting your journey through the disease. But this may explain why some patients have more serious breakthrough infections than others that's not resolved. We also found that these autoantibodies, especially interferon alpha, but the other ones as well, tended to associate with these gastrointestinal and some of these sort of respiratory viral-like PASC symptoms. And so, and finally, we found that if you had autoantibodies when you were at convalescence, you pretty much had autoantibodies at diagnosis, and they were mature. There were no IgMs, they were all IgGs. And so the suggestion is that even though patients may have autoantibodies that are subclinical, meaning that there's no maybe medical history that would associate or indicate any problems of these autoantibodies, that a number of patients do have them, and that they have an impact on PASC, and it's actually a pretty strong impact, and we'll talk about that a little bit more. And when you look at the patients that have very high autoantibody levels, they tend to actually have higher of these extrafollicular, these atypical memory B cells. Once again, suggesting that these autoantibodies may actually have a functional impact on the patient's immune journey through the disease. And so that was when I talked before that atypical memory B cells may just be associated with an infection. True, but if you have these autoantibodies associated with lupus, you actually have more of these atypical memory B cells. So that both of these views from the literature seem to be true. Okay, so we also did single cell analytics at quite a deep level. And I'm just going to talk about a little bit about what we found here. And this is work that we did also with help from Adaptive Bio and our own work to try to understand the specificity of T cells against the virus. And so what one expects to see in patients over time is that you would have clonotypes from a naive pool that would expand during peak disease to form effectors. And then these would contract into a sort of a memory pool. And so when we looked at these patients, here now you've got the single cell RNA-seq UMAP presentation that I think many people are familiar with. And you can see this red here is effector. And then you've got sort of a naive-like phenotype here, and then the stuff in between. Here's where the SARS-CoV-2 specific T cells tend to sit, largely in the effector, but also you see them in this very small subpopulation, which is the what are called proliferative exhaustive phenotype. They're basically cells that where all the proliferation is taking place, but they express all kinds of markers. Okay, so in most patients, you see these effector populations of SARS-CoV-2 specific T cells drop over time. Not in patients that exhibit gastrointestinal PASC. And if you look at CMV specific T cells, and remember there's no CMV specific reactivation that we can find, you also find that those decrease over time in the infector pool. But here, there's the data, not in patients that show gastrointestinal PASC. And so what this tells us is that GI PASC seems to have a slightly different kinetic process for development. You can't really anticipate it unless you're actually following these T cells that these effector populations that tend to increase over time. And that really differentiates the patients that develop GI PASC from other patients. And it doesn't matter whether SARS-CoV-2 or CMV specific. And so it argues that there is just a generic activation of immune system, of T cells in the immune system against in patients that have this type of GI PASC. Okay, so we have these PASC factors that most of which can be measured at diagnosis, with the exception of these T cell populations. There's the autoantibodies, there's type 2 diabetes is the dominant comorbidity, and then there is the EBV viremia and SARS-CoV-2 RNA-emia. And so we took these various PASC factors and I not labeling everything we looked at here, but the ones I just the autoantibodies, viremia, diabetes, et cetera. And then we tried to associate them and understand, are they related or are they non-related, by looking at their correlations across this pretty large data cloud of other measurements that we've done. And what we found, I think, is very instructive in terms of understanding the nature of long COVID. So at diagnosis, one sees actually a lot of interrelationships between these autoantibodies. You see a little bit of interrelationships between viremia and diabetes. Once you go to acute disease, you still find these relationships, but they're a lot weaker. And once you go out to convalescence, they're practically non-existent. You can't see them. And what that argues for, it's basically, if you think about, an example might be in physics, chaotic systems, is that at the initial starting point, you have a lot of, a limited number of pre-existing conditions or existing conditions that can lead to long COVID. They're related. You might have a handful of treatments that you would suggest by looking at this stage. But over time, because everybody starts slightly different, they're different age, different comorbidities, different sex, et cetera, these relationships just are lost. And out here, you would take a look at these patients and you would say there's almost a different treatment per patient, which is probably not true. This is probably more accurate disease, a picture of the nature of this. And I think this really goes to explain why these long chronic diseases are so challenging to understand. Because out here, most of what people might think of as causal relationships are actually lost, but this is at the time when patients are presenting with these conditions. Okay, so in conclusion, each one of these PASC factors, viremia, RNAemia, type 2 diabetes, explains roughly a third of the PASC that we found. With the viral measures, more associated with the neurological PASC and type 2 by diabetes with respiratory viral PASC. And then the autoantibodies explain about 2/3 of the PASC. So some patients had multiple ones of these PASC factors, which is why it adds up to more than 100%. But the autoantibodies tend to be the most important. Now, some of the important notes on this autoantibodies is that when we measure these, we are doing quantitative titers. If you went to, say, LabCorp and had autoantibodies measured, they're only going to give you a cutoff level of what is considered a clinical level of autoantibodies. And so you're not going to capture most of these patients. You would capture the ones at the very high levels. But from what we find is that if you have autoantibodies, they actually can play a role in determining PASC. It doesn't, their level is important, but it's not fully deterministic. And then, limitations of the study, which we're, working hard to address right now, is that, we're only looking at a few months post-diagnosis. We're only at 300 COVID-19 patients. And so if you're trying to look at one PASC factor, say women, that's actually a pretty small number of patients.
James, we're at the 2-minute mark.
That's fine. This is I think it's my second to last slide. And so you're not going to really have a statistical significance. And this is one reason why the National Recover Study, which we're doing a big chunk of, is trying to look at 15 or 18,000 patients. Type 2 diabetes is resolved simply because of the numbers. We only looked at a handful of autoantibodies. We're now trying to interrogate the full proteome of autoantibodies with Mike Snyder. We aren't sensitive to PASC from the Omicron variant, and surely it's going to be different because of the infection routes. And, one would guess it might be less, but who knows? And then I'm always looking for talented postdocs. So if you want to contact me, there's my email. And I need to acknowledge and be very grateful to a number of people that stepped up and helped us do this study. Thank you.
Thank you very much, James. I noticed that Francis Akita has their hand up. I'm not sure if they want to talk, but if so, you can unmute yourself. Or that may have been an accident. So Zizeng Sheng has a question, and you should be able to unmute yourself.
Hey, I'd like to talk. So basically, the one question is you saw the EBV connection with COVID. In the past, we know EBV antigens like EBAA1 also elicits antibody that's cross-reactive with the anti-nuclear autoantibodies. So basically, those autoantibody anti-nucleus antibody could be elicited by the EBV antigen. Have you seen any connection in the COVID cases after the EBV activation? There's those kind of EBN1 antigens could lead to autoantibody production.
I think that's a very good question. I mean, I think in most of these patients, as I indicated, we do have they have autoantibodies prior to the, prior to getting infected by SARS-CoV-2, or they wouldn't be IgGs. But your question is very valid. And right now we are taking all the blood from patients that had these autoantibodies and had EBV, and we're working with Scott Boyd to go through and completely characterize the B cell, the B cell receptors and the autoantibody sequences that we're seeing to understand and address the question you're asking. I don't know the answer yet, but it's a great question.
Do you happen to know any mechanisms for the EBV activation in COVID?
EBV is largely sequestered, from what I understand, in B cells.
Right.
So it may simply be tissue damage, or it may be B cell activation that can lead to that. But we did look for EBV reactivation in patients that received the virus to see if maybe just simple EBV, simple stimulation of the B cells could cause it, even if it's transient and low level, and we don't see it. So my guess is it's probably tissue damage, but there may be someone on this call that knows more than I do.
Okay, thanks.
James, if I could ask a couple of questions. Can you comment quickly on the potential relationship, if any, between long COVID and other types of long syndromes? So for example, you mentioned syndromes associated with chemotherapy and Lyme disease. And then my second question is, I think the answer is no, but do you have enough information to make an a priori prediction on what patients are most likely to develop long COVID? And if not, do you think it will ever be possible to do that?
Well, okay, so there's two questions here. The first one,
the relationship to other long
exactly. Okay, so we did, in fact, over the past year, looked at a bunch of oncology patients, females with ovarian and breast cancer undergoing radiation chemotherapy, to understand if chemobrain could be related to the kind of things we see here. And you do actually see quite a few autoantibodies that develop and under treatment, but they're all IgMs that don't seem to mature. So if there's a relationship, it's not a low apple to trying to resolve it. I would guess that post-acute Lyme is another one that we've been doing quite a bit of work on. I think the relationship is likely to be a lot more direct in terms of its connection. I think the challenge is that having the serial draws on Lyme patients on the sort of quality of biobanks that are required to do this kind of study is still a little bit challenging. We had a Lyme study we were starting right before the pandemic. I would do it completely differently now based upon what we learned here. So I think that's not resolved yet, but I'm fairly certain, I would bet money that is going to be strongly related. And I would guess post-acute sepsis conditions may have some similarities as well. And then your second question is, well, if for sure, you could, type 2 diabetes patients, that ahead of time. If you, if we looked at these patients that reported, say, 3 or more long COVID symptoms, and that's basically, of the patients that report any symptoms at all, it's basically most of them. I think it's like 2/3 of the patients that report any symptoms. And we asked, do they have one of these 4 PASC factors: autoantibodies, EBV viremia, SARS-CoV-2 RNAemia, or type 2 diabetes? every single one did. I think we quoted it as greater than 95%, but there were no exceptions in our data set. And so that does argue that you can anticipate this. You could know about it ahead of time in most patients. Now, there's some conditions that we are not sensitive to, like, people lose their hair. There's cardiac issues that are more rare that we don't resolve in the size of our data set. So it's probably not universally true, but it seems to be for the types of symptoms that we were looking at here and we're able to resolve. Yeah, you can see it ahead of time.
Okay, we have one more question from Stuart Firestein. Stuart, you should be able to talk.
Great, thanks so much. I'll make this very quick. I'm just wondering from a treatment point of view, whether any treatments are being tried that are successful that would suggest what the underlying causative factors are. I'm thinking in particular of the use of drugs like modafinil, which are common in post-chemotherapy treatments for fuzzy brain sorts of neurological issues. Do you know anybody using those sorts of treatments in these cases?
I haven't seen it. I know that, antivirals are sort of an obvious one that's suggested by this early in the disease course, but of course, those are being used anyway. And so my guess is epidemiological studies will resolve whether those are effective or not. I know that at least in some of the hospitals around here, people we've been working with have been trying cortisol replacement therapy and for some of those patients that were are found to be cortisol deficient. I haven't heard about any of these other treatments. I think, I'm sure they're being tried, but I don't know. It's still pretty early days. I think the NIH just put out a call to try to integrate therapy treatments with their large recover study. And in fact, if there's anybody who wants to do that, please reach out to me, because we are more than happy to try to incorporate such studies with our patients.